This video demonstrates a procedure to quantify forepaw dexterity during food handling in rodents. Each animal is exposed to short lengths of uncooked pasta in first the home cage and then the testing environment. In the testing environment, the animal's forepaw movements are videotaped during pasta eating. The adjustments made with each forepaw are identified and counted during slow-motion video playback. During the video analysis, atypical handling behaviors are identified. Results show changes in forepaw dexterity after brain damage or during aging through the quantification of movements used to manipulate food during eating.
Hi, I'm Tennant from the laboratory of Theresa Jones in the Department of Psychology at the University of Texas at Austin.
And I'm Asay, also from the Jones lab.
Today we're going to show you procedures for the Vermicelli handling test in rats and the Capellini handling test in mice.
We use these procedures in our laboratory to study how dexterous forepaw use during food handling changes after brain damage or during aging.
So let's get started.
To prepare the pasta pieces for testing, use an ultrafine tipped marker to mark DeCecco brand Capellini and Skinner brand Vermicelli at specific intervals with a cardstock template. For Capellini, use 0.86 cm increments, and for Vermicelli, use 1.75 cm increments. This facilitates visualization of the movement of the pasta strand during eating. Using a razor blade, cut Capellini to 2.6 cm lengths for use with mice, and for rats, cut Vermicelli to 7 cm lengths.